Good morning, all of you.
Good morning, sir.
Good morning, ma. Okay. In the last class, we have discussed some of the concepts related to water, plant water relations. There is water potential, solute potential, pressure potential. Okay. Today, we'll discuss about some of the concepts related to this. Okay. That is osmosis. Okay. Osmosis. Okay. What is meant by osmosis? Okay. When you observe here, we all know very all the living cells are covered by the membrane called as the cell membrane or plasma membrane. Okay. Okay, like that in case of plant cells also, the plant cell is surrounded by a cell membrane and a cell wall. Okay, the outer covering is the cell wall, and inner to the cell wall we'll find the plasma membrane or cell membrane. So that is, so the plant cell is having two structures, that is cell membrane, and outer to this cell membrane we'll find the cell wall. Okay, that is, the cell wall is freely permeable to water. The cell wall is freely permeable to water and substances in solution, hence is not a barrier to movement. Okay, while we are discussing about the types of membranes, we have come across this permeable membrane. That means which will allow substances to pass through it. It may be the water or solute or substances or whatever it is. Okay, one such membrane is or one such structure is the cell wall. So the cell wall is freely permeable to water and substances in solution. That is, whatever the substances that are dissolved in the water are also get transported. Are also get transport. Okay, are also get transported through the cell wall. Because that's why the cell wall is not considered as a barrier. Okay, that is, it will, because it is allowing all the substances to pass through it. And next, in plants, the cells usually contain a large central vacuole whose contents, called as the vacuolar sap, contribute to the solute potential of the cell. Okay, when you observe the plant cells, very important characteristic feature of a plant cell is presence of a large central vacuole. Large central vacuole, and this vacuole is surrounded by a membrane called tonoplast. Okay, large central vacuole which is surrounded by a membrane called tonoplast, and the okay, and the fluid that is present inside the vacuole, we are calling it as vacuolar sap. Okay, which will contribute to the solute potential of the cell. That means okay, whatever the components that are present in the vacuolar sap, that one will determine the solute potential of the cytoplasm of the cell. So that one will determine the turgidity of the cell also. That's what you have to remember. In plant cells, the cell membrane and the membrane of the vacuole, that is the tonoplast, together are important determinants of movement of molecules in or out of the cell. So the membrane that is surrounding the cell, we are calling it as cell membrane. And the membrane that is surrounding the vacuole, we are calling it as a tonoplast. Okay, this is cell membrane and this tonoplast are very important membranes. Okay, they are considered as the semi-permeable membranes. Okay, which will determine the movement of molecules in or out of the cell. Okay, which molecules has to enter into the cell and which molecules has to move out of the cell, that is determined by this cell membrane and the tonoplast. That is the membrane surrounding the total cell and the membrane that is surrounding the vacuole, that is the tonoplast. These two membranes are selectively permeable membranes, and they will only determine the movement of molecules across the membranes, which molecules has to enter and which molecules have to leave the cell. So, now the question arises, the osmosis. Osmosis is the term used to refer specifically to the diffusion of water across a differentially or selectively permeable membrane. Okay, that means here osmosis means the movement of water. Okay, in osmosis we will not discuss with related to the movement of the solute particles. In osmosis we'll discuss with related to the movement of water or diffusion of water across a differentially or selectively permeable membrane. Differentially selective, okay, selective membrane or a selectively permeable membrane. Okay, that means they will allow, that means the membranes which will allow only certain molecules to pass through it. That membrane we are calling it as a differentially permeable membrane or selectively permeable membrane. So here in osmosis, the term osmosis refers to the movement of water or diffusion of water across the membrane, selectively permeable. Okay, that is. And next, osmosis occurs spontaneously in response to a driving force. That means the movement of water in osmosis occurs according to the water potential, according to the gradient, according to the gradient, okay, concentration gradient of water. Okay, that is. So that concentration gradient of water only drives the water to move from one side of the membrane to the other side of the membrane. That is the driving force. The net direction and the rate of osmosis depends upon the pressure gradient and the concentration gradient. Okay, that is. That means when you here, one thing you have to remember, ma. Okay, when you observe here, for example, if you take a beaker which is separated by a selectively permeable membrane, which is separated by a selectively permeable membrane. Okay, and we have taken two different solutions. Okay, for example, here we can say no. Okay, A side and B side. A side is having 2% solution and B side is having 5% solution. Okay, when you observe here, okay, from where to where the water will move? From A to B or B to A? Can anyone say? Okay. Okay, first of all, which region is having more water potential? A side or B side?
Yes, sir.
B is having more water potential.
A.
Okay. How the B will be having more water potential? Okay, when you add any solute, the water potential will be decreased,
Yes,
no?
sir.
Okay, which is having more solute? 5% is having more solute, no? Automatically that B will be having less water potential, no? Okay, one thing you have to remember, ma. Okay, the water movement will occur from A to B and B to A also. but the net movement will be, okay, when you are separating, that means here one thing you have to remember, the water, okay, in osmosis the water will move in both the directions. If a okay, if you take any two sides which is separated by a semi-permeable membrane, through this semi-permeable membrane the water will move in both the directions. That is, from here, when you take here A to B and B to A, in both the directions the movement of water will occur, but the net movement, the resultant movement will be from A to B, because A is having more water potential and less concentration. So that's what we are saying. The osmosis, okay, that is the statement that we can see very clear. Okay, the net direction, that means the movement of the water will occur in both the directions, A to B and B to A. But the net direction, that means overall, okay, when you observe from where to where the water will move, means that rate of osmosis depends upon the pressure gradient, that is okay, the water potential, and a concentration gradient, that is the solute concentration. Okay, where there is more solute concentration, there less water potential or pressure gradient, and where there is low solute concentration, there will find a high water potential. So the water will move from the region of higher water potential to the region of lower water potential as a net. Okay, that is, so the net movement of the molecules, okay, the net movement of water molecules will occur from a higher water potential to the region of water potential, or in other terms we can say that from the region of lower concentration to the region of higher concentration. Here we refers to the solute concentration. In the first case, we refer to the concentration of water with reference to water potential. Okay, that is, so that is what you have to understand. Understood? Anybody is having any doubt regarding this? Okay, that is, the net direction and the rate of osmosis depends on both the pressure gradient and the concentration gradient. Here the pressure gradient refers to water, concentration gradient refers to solute. Okay, that is, so water will move from its region of higher chemical potential or concentration to its region of lower chemical potential until equilibrium is reached. Okay, until equilibrium is reached, until the equilibrium is reached, okay, the movement of the water will occur in both the directions. Once it reaches to the equilibrium, the net movement of water molecules, that means the water molecules will move from A to B and B to A, okay, up to the okay, even in the equilibrium also the movement of water from A to B and B to A will occur, but the net movement is zero. So how many number of molecules of water is moving to B? The same number of molecules will move from B to A also. So that's why we are saying that the net movement will be zero at equilibrium. Yes. Okay, in this case, we can take like this as, okay, 5 plus 2, 7. So if both the, okay, 5 plus 2, 7. So if the concentration on both the sides reaches to 3.5%, because here what is happening, okay, In A region, 2% is there, and in B region, 5% sugar solution. And now what happened? The movement of water will occur from A to B. The net movement of water will occur from A to B. So slowly what happens? The concentration of A increases and the concentration of the B slowly decreases. And finally, what happens? There is some point at which the concentration of both A and B becomes equal. That is, here it is 3.5. That point where we'll find both the concentrations become or water potential becomes equal, that point we call it as equilibrium. At that equilibrium point, the net movement of water molecules will be zero, because how many number of molecules from will move from A to B? The same number of molecules will move from B to A, but before, more molecules are moving from A to B. As a result, the A concentration is increasing and B concentration is decreasing because of the addition of new water molecules. So at one point, the concentration of both the sides becomes equal, and at that point we are calling it as equilibrium. At equilibrium, the net movement of molecules is zero. Okay, that is what we can see here. Okay, there is a initially there is a difference in the concentration or difference in the water potential. Okay, the water will move from one region to the another region, but after certain period of time, the concentration becomes equal on both the sides. So we say that the the two sides have attained the equilibrium. Okay, at equilibrium, the water potential of both the sides is equal, whatever it may be. It depends upon the concentration, the value of both the sides. Okay, that is okay. And next, you may have made a potato osmometer in your earlier classes in school. Okay, if a potato tuber is placed in water and water enters the cavity in the potato tuber containing a concentrated solution of sugar due to osmosis. Okay, in your lower class, you might have studied, okay, you might have conducted these experiments, I think in ninth class. Potato osmometer. Okay, in potato osmometer, what they will do means they will, they will take a potato, they will peel the outer layer because the outer layer is covered with a cuticle. The cuticle is impervious to water. So that's why they will peel the cuticle, the outer layer of the potato. And now what they will do means they will scoop. Okay, that means they will make a small pit, okay, inside the potato by peeling, okay, by scooping, by using either some structure, they will scoop and they make a small pit in the potato, and in that pit they place the concentrated sugar solution like 2% or 5%, like that. And next they will take a beaker, and then in that beaker they place water with low concentration. And now the potato will act as a semi permeable membrane, because the potato is a living and it is consists of cells, living cells, and that the cells of the membranes of the cells in the potato will act as a semi permeable membrane, and through that the water that is present in the beaker, okay, through potato, which is acting as a semi permeable membrane, they reach the pit of the potato where the sugar solution is present. Okay, you can fill that pit with a half with the sugar solution. After some time, you can observe there is a rise in the water level, and if it is continued, we can observe after some time we can observe there is an overflow of water, and this one will be continued till the concentration of the water that is present in the beaker and the concentration of the sugar solution that is present in the pit of potato reaches to equilibrium. Okay, reaches to equilibrium. Okay, that is called potato osmometer. That what you have studied in your. Okay, here it is given one diagram is given ma. Okay, study the figure 11.3 in which the two chambers A and B containing solutions are separated by semi-permeable membrane. Okay, here the some questions were asked. Solution of which chamber has a lower water potential? Here, okay, okay.
B.
Now what? Yes, ma. Okay, first you have to, okay, in both these surfaces, the amount of water is same. Amount of water. We are not saying the water potential. We are saying amount of water. For say both are having 100 ml or 200 ml of water. Okay, both the sides are having 200 ml of water. Say, but the number of solute molecules, because the water potential depends on the solute potential. Okay, the number of solute molecules that are there. Okay, the more the number of solute molecules, the less the water potential. Okay, that is, we can observe in the chamber A 1, 2, 3, 4, 5. Only 5 solute particles are present. But in the chamber B, we can observe more than 5. Okay, 5, 4, 9, 11, 12, 13, 14, 15, 16. 16 solute molecules are there. So, which is having more concentration, A chamber or B chamber? More solute concentration. Okay, which chamber is having more solute concentration? A chamber
B.
or B chamber? B chamber. So, more the solute concentration, the less the water. The
Water potential.
okay, the water potential of a water will be gradually decreased when you add, okay, when you are adding the solute particles. How much you are adding the solute particles, that much is the decrease in the solute potential. So, as the B chamber is having more solute molecules, automatically that B chamber will be having the lower water potential. And next, the second question. Solution of which chamber has a lower solute potential? Okay, solution of which chamber has a lower solute potential? They
A.
asked related A chamber, because in the A chamber only 5 solute molecules are there. So the solute potential will be less. Okay, that is. And next, in which direction will osmosis occur?
A to B,
A to
sir.
B. Because he is having a less solute potential, more water potential. Whereas the B chamber is having a higher solute potential, lower water potential. Always in osmosis, the water will always move from the region of higher water potential to the region of lower water potential. So the water will move from A chamber to B chamber. The net movement of the water molecules will be from A to B chamber. Okay, that is. And next, okay, the next question. Which solution has a higher solute potential? Okay,
B.
okay, higher solute potential, that is B. Yes. Which solution has a higher solute potential? Okay, the solution which is having solute particles, that one will be having a more solute potential. Okay, that is a B chamber. Okay, that is. And next, next question. At equilibrium, which chamber will have lower water potential?
Sir, both will have same.
Yes.
Equal.
Okay. At equilibrium, both the chambers will be having the same water potential. There is no change in the, or there is no difference in the water potential between the two chambers at equilibrium. So the question is wrong. No, no. Okay, okay. We have to say no, and we have to answer at
Both
equilibrium.
are
Both the
equal.
chambers are having the same water potential. Okay, and next the next question. If one chamber has a water potential of 200 kilopascals and other is having 1000 kilopascals, which is a chamber that has the higher potential? Okay, which is having the higher potential? Okay, if one chamber is having 200 kilopascals and other is having 1000 pascals, which is the chamber that has the higher water potential? Okay, obviously that is 200 only, no? We are applying
No.
pressure, ma.
Yes, sir.
Okay, where we are applying more pressure, there the potential will be more, no?
Sir.
Okay, so 200. Okay, the chamber with 200 kilopascals will be having more potential, more pressure potential. Okay, and next, what will be the direction of movement of water when two solutions with water potential 0.2 pascals and water potential of 0.1 pascals are separated by a selectively permeable membrane?
Sir, high to low.
Okay, that is high to low, that is 0.2 to 0.1. Okay, here the pressure, okay, pressure is given. Okay, now they are exerting some pressure. So the pressure, okay, where there is more pressure, there more pressure potential will be there. So water will move from higher pressure potential to the region of lower pressure potential. Okay, that is. Okay, next, let us discuss another experiment where a solution of sucrose in water taken in a funnel is separated from a pure water in a beaker by a selectively permeable membrane. Okay, there is, okay, that is, you can get this kind of membrane in an egg. Okay, how to get this egg membrane? Okay, for that what you have to do means, okay, take the egg of a hen and place the egg in a dilute hydrochloric acid. Okay, when you observe here, the shell of the egg is made up of which material? Okay, the shell of the egg is made up of which material? Calcium carbonate. When you place it in a dilute hydrochloric acid, it get dissolved. Okay, we have to keep it for a half an hour or 2 hours. Sorry, 1 hour or 2 hours. Slowly, the eggshell get dissolved in that hydrochloric acid, and the egg membrane is exposed, or the membrane surrounding the egg is exposed. Is exposed. Now, what you have to do means, okay, okay, make a small hole. Okay, carefully remove all the yolk from the egg membrane. Okay, whatever the yolk that is present inside the yolk egg membrane, remove all the yolk, and now you will get the egg membrane, that which will act as a semi permeable membrane. And now what we have to do means, you have to take a funnel, say thistle funnel, they say it as thistle funnel, like this. Okay, with a long narrow end and a broad opening, and the broad end of this thistle funnel is covered with this egg membrane, and it is tied tightly without leaving any gap. And now what we have to do means, the thistle funnel with the egg membrane is filled with a concentrated sugar solution, highly concentrated sugar solution, say 5%, 10%, like that. Okay. And now what we have to do means, we have to take a beaker, and in that beaker you have to place the pure water, say distilled water or pure water, in which the water potential is 0. Okay, now the thistle funnel with the concentrated sugar solution is placed in the beaker contain pure. Now observe the change that is observed in the thistle funnel or in the beaker. Okay, when you observe, we can observe there is a gradual increase in the sugar solution, level of sugar solution in the thistle funnel. Okay, this is the experiment which is similar to that of a potato osmometer. How the water is entered from the beaker into the, okay, that is so how the osmosis here, the potato is acting as semi membrane. In the same manner, the water entered through the semi permeable membrane that is placed at the broad end of the thistle funnel. Okay, will act as a semi permeable membrane, and through that membrane the water enters into the thistle funnel from the beaker. Gradually there is an increase in the sugar solution. Okay, this process will occur till the concentration becomes equilibrium. Concentration reaches to equilibrium. Okay, that is okay. And next, there is a continuation concept is there, that one we'll discuss in the next class.